Thanks for coming to our 5th summer lecture in this year's annual series. Hard to believe we're almost through the summer in terms of lectures. And next week I want to remind you that our final lecture in the series will be presented at this same time and place. Christian Kieslowski from the National Center for Electron Microscopy will be our special guest speaker, and he will talk about if we could only account for every single atom. So I hope to see you here next week at noon. So now I want to bring up here Rolly Otto, who is the manager of the Center for Science and Engineering Education and one of the founders of this lecture series, and he'll introduce our speaker. Rolly?
Thank you, Ron. It's my privilege to be able to introduce the speaker to you today, and you know, I've known Ellie Blakely for a number of years now because she has been a mentor for our undergraduate students and even our high school students, and with her and her team. But I just now really got to know and realize that how long she's actually been at the laboratory and what her career has been all about. So let me introduce you to her a little bit in the way that I've kind of learned about it just recently. Ellie got her bachelor's degree at the University of San Diego, and went to the University of Illinois, Urbana, to get a master's degree and eventually a PhD. Now, interestingly enough, as an undergraduate, like many of you who here at Berkeley Lab, she worked at Oak Ridge National Laboratory in her junior year, and did some radiation biology studies there. So in this way, she was introduced to her future career. She also worked at Argonne National Laboratory, one of the other big multi-program national laboratories, and working on her PhD under a professor at the University of Illinois, Urbana. And from that, then, she was hired in 1975 to be a staff biophysicist here at Lawrence Berkeley Laboratory, and she worked with one of the founders of our life sciences radiation biology area, and that was Cornelius Tobias. Today, she is a senior staff scientist, biophysicist in the life sciences division. She holds 2 adjunctive positions with the other universities, one at Loma Linda University, where she's a clinical professor of radiation medicine, and another, an affiliate appointment with the Department of Radiological Health at the Colorado State University, Radiological Health Sciences, that is. She's an appointed member for the National Council on Radiation Protection and Measurements, the Scientific Committee of Guidance on Radiation Received in Space Activities. She says that's the low Earth area, and that's just coming to an end, or has come to an end. But she's been also appointed as a member to the Scientific Committee of the National Council on Radiation Protection and Measurement for deep space work. The name of that committee is the Protection Recommendations for Travel Beyond Low Earth Orbit. She's particularly proud of the fact that she was an advisory committee member on radiation quality and radiation biology in hadron therapy. This is for the International Atomic Energy Agency. Recently did that kind of work. And one of the things that Ellie was recognized for just recently was the Secretary of Energy Outstanding Mentor Award. So with that, I introduce to you Dr. Ellie Blakely.
I'm very honored to have this opportunity to tell you about the work that I've been doing here at the lab for almost 30 years. It's actually gotten to be a very interesting story the way it has intertwined, and so I hope to share that with you today. The scope of my presentation, in order for you to appreciate what we've done in the science, I need to give you a little background in history, and then I'll tell you about some of the science that we did and are currently still ongoing with, and then give you a view for the future. For NASA in particular, one of the challenges of space exploration is an understanding of the biological effects of exposure to relatively low doses of a very complex radiation field in an environment of additional stressors like microgravity, which is true in space. And just like any employer, they must follow OSHA rules and be safe for their occupational exposures of their employees. So to acquaint you with the hazard of radiation in space, I want to start my story with Marie Curie. This is a picture of her in 1913. She, to me, was a pioneer. She's the only woman to hold 2 Nobel Prizes. And the first one was for work she did in the discovery of plutonium in 1903. And this is one of the cloud chamber pictures that she obtained, which shows the whole story of my presentation today. It shows that not all radiations are created equal. And what you see are alpha, these thick, heavy, straight tracks of ionizing radiation, and beta, these wiggly, thin tracks. And the fact that they, passing through biological material, cause quite different effects is really the story in space. Now, the cloud chamber that Mother Nature has is another way you can illustrate very graphically radiation, and that's the Aurora Borealis. This happens to be the southern Aurora Borealis near Australia. What you see is there a spacecraft at the edge overlooking the globe, and you see these parallel lines of radiation. And what it actually represents, as illustrated in a recent National Geographic in November of 2001, is when the plasma from the cosmic radiation interacts with the atmosphere, you get to actually see the condensation of the radiation. Where does the plasma come from? Well, in the cosmos, we have solar events where solar wind is generated from the sun. And I have these photos courtesy of NOAA in Boulder, Colorado. What you can see is the sun is blocked out because it's so bright. And what you see are these large solar flares emanating many miles out of the sun. And those particles traverse through the void of space and come to our atmosphere. And it turns out at the top of the poles, the North and South Pole, there's a defect in our magnetic field. And that's why only there the radiation comes in and can be visualized in the aurora. Now, we have a wonderful support from NOAA because when these solar events happen, and they cannot be predicted, they really are not able to predict it, but you actually see quite high fluences of protons of various energies emanating from such an event, and they last many days and hours. So this is a set of measurements from a very large event that took place in October 1989. And it also was coincidental with measurement of neutron peak in a deep cave. So we can actually characterize the kinds of radiation in space from Earth, but we need to really understand what a crew member on a way to Mars might encounter. And the total picture of different kinds of radiation in space are illustrated in this presentation, where the transport of all of the different components on this log-log scale of flux versus particle energy shows you the solar wind up here, protons at the upper left, the trapped electrons, the trapped protons, solar protons, and they cover massive range of energy and effectiveness. There also is a component from the galactic cosmic rays that are heavier than protons. And behind shielding in a spacecraft, estimates have had to be made in order to protect the occupants. And what has come out of that work is the fact that there are many species of high Z elements, including iron in particular, has a high peak. And so one of the things, because of this high effectiveness of iron, that's a major concern for shielding. Now, I mentioned high ZE, so I thought I should define it for you. The Z, of course, is the term we use for particle charge, and the E is particle energy, and they refer to this as high just because of the energy range that I spoke to you about. But if you know the Z and the E of a particle, then it actually defines the ionization quality of the track and the particle irradiation. What do these tracks look like? Well, I've already shown you Marie Curie's tracks, but what does an iron ion look like? It's very heavy. Here's an iron 56 track with a delta ray coming off tangentially, and this can do a lot of damage depending on where it passes through the body. Let me just mention a few definitions of units. The scientific units for radiation absorption have changed. We used to call things rads and rems, but now we call them grays and sieverts. And I just wanted to briefly tell you what the difference was. A gray tells you the total ionization, but it doesn't tell you the effectiveness. And that's why they invented the word sievert. So mostly you'll see me speaking of sieverts because it takes into account the different effectiveness of different ionizations. Also, the sievert and the gray are big units of dose. Those of us who are radiation workers actually have a limit that we're allowed, to, quote unquote, to get, and obviously as low as possible would be preferred. But the limits that are allowed, actually, we even have it lower administrative limits at our institution here. Now, in addition to the high effectiveness of the track, the actual absorption as a function of depth is quite different for these particles compared to low LET photons. Photons, as you step away from them, the dose is dropping off exponentially. But these particles do just the opposite. As you get to the stopping point, they dump their energy. And here in a parameter we call LET, the linear energy transfer per unit track, there is a maximum absorption right as the particle stops. And this is called the Bragg peak, a very important concept in particle physics. Now, Ernest Lawrence, the man for whom our lab was named, was very important for his invention of the cyclotron. And what the cyclotron allowed was a creation of this radiation that we see in space, but being man-made here on Earth, to allow biologists like us to study it. He also won a Nobel Prize in 1939 for his invention. His first cyclotron was so small it would fit in your hand, but they grew. And he also engaged his brother, Dr. John Lawrence, who was a physician, to join him in using these unique properties of these kinds of radiations for medicine. Excuse me. So now I'm showing you an aerial view of our laboratory in the 70s. And it looks quite different now because there have been new buildings and construction and vegetation. But I just want to use it as a tool to tell you about 4 major accelerators which were operating when I first came here to the lab. You'll see some round buildings like the Cal Stadium, that's not the one. And you can see the Lawrence Hall of Science, that's not the one either. But there is this very notable dome which is currently housing the advanced light source. And when I came, there wasn't an advanced light source. It was called the 184-inch, because of the size of the D, accelerator. And so that was a premier accelerator for helium ions, and we actually used it to treat eye tumors. And I'm going to tell you a little bit about that story, because that's how I got into cataract. In addition, this prominent machine here was called the Bevatron. And the Bevatron actually had an injector from the Supra Hylac. Dr. Al Giorso had a very clever idea. The Bevatron couldn't get to the energies we needed, so he said, let's link this accelerator to this accelerator and make very high energy. And actually, this machine, during the time before '93 when it was closed, actually accelerated particles of iron for NASA programs and clinical programs. We're using lower atomic number particles like carbon and neon. And that work, which is now called hadron therapy, is ongoing all over the world, especially in Germany and Japan. And it's a whole field that's showing that the physical distribution and properties of these radiations do have medical benefits, primarily because they restrict the dose to normal tissue and only limit the dose to the tumor. In addition, I want to draw your attention to one rectangular building which is still operating, and that's the 88-inch accelerator. And I'm very grateful for that machine, because now, compared to all of these others, this is the only one that I now can still use here on the site. And it's been a very wonderful source of protons and nitrogen ions for some of our studies. Now, I need to tell you a little bit about the anatomy of the eye for you to appreciate what I'm going to tell you. Unfortunately, this was an eye that was lost due to a large ocular melanoma, this ugly thing here. And the reason the eye had to be removed was not so much because the tumor couldn't be treated, but because it detached the retina. This flap here is the detached retina, and that required, due to the bleeding, that the eye be removed. Now, the anatomy I want to draw your attention to is the lens. In the adult, the lens is avascular. It doesn't have a blood supply. And it is nourished by cells on the inside of the cornea. This is the cornea. And it's a bilayer of cells, and epithelium on the outside where people who wear contact lenses hang their contact lens. And in the inside, there are endothelial cells. And I'm going to come back to the story of the endothelial cells. But what we found was that the precision with which you could use these particle beams to treat such a tumor. Now, in this orientation, what I'm showing you is a CAT scan of the eye. This white is the bony structure at the side of the bone here, the eyeball. And the eye is oriented so the lens this time is upright. You see the structure of the lens. This again is the tumor. And what are these rectangular boxes? Those are called isodose contours, and that was the dose to the tumor and adjacent metastatic disease. The hot colors, the red, represent 100% of the dose profile that the physician prescribes. The cold colors, the blue here, represent only a 10% isodose profile. And so the beauty of these treatments is that none of the normal brain behind that 10% dose received any other radiation. So it's very precise and it's restricted to the region of the tumor. However, depending on the location of the tumor relative to the lens, you would get additional secondary effects. And I'm sorry to change the presentation of the eye. This is a flat presentation of the eye. Let me just orient you. The red thing is the tumor. The yellow discs represent tantalum rings that are sewn by the eye surgeon on the back of the sclera to help us align with X-ray exactly the location of the tumor. This green circle is the lens. This is the optic nerve. And these structures are here because the physician knows if he irradiates the optic nerve or the lens, he might get effects he doesn't want. Now, what you can't tell from the flat presentation is that this tumor was located very far in the back posterior region of the eye. So the lens could be spared. And as you see, the dose volume histogram here to the lens shows protection, even though the dose to the tumor was 100% as the doctor prescribed. The lower panel shows a much different situation. Unfortunately, this patient waited much too long to go to the eye surgeon and the eye doctor. And unfortunately for them, the eye tumor was located in the anterior portion very near the lens, and so they could not spare the lens. So while I was working as a biologist with this clinical program, I realized we had, in fact, a human dose response to lens, in that some of the patients sustained a high dose and others a lower dose. So I thought this was important, and the physicians were a little concerned about me. They said, let me get this straight. We cure them of their eye tumor, they get to keep their eye and they have vision, and you worry about a plastic lens being installed? And I said, no, the point is that this is a late effect of radiation for which we have a very graphic endpoint that can be quantitated, and I think it's important to study. Now, the treatment of eyes with particle beams, the helium here, this only shows out 10 years, but we actually have more than 25 years of follow-up for the survivors that lived that long, is very successful, 97% local control, and with few complications. So this was a very successful treatment. But I coerced a young resident at the University of California, San Francisco Ocular Ophthalmology Department, Will Meekum, to do a study with me of the patients, looking at the percentage of lens in the field and the incidence of cataract, because so many of them got a cataract. We published this in Archives of Ophthalmology in '93. What we found was, and these curves are hard to read if you're not familiar with them, because you have to take away the fact that in life, many of us do get a cataract for other reasons, so you have to subtract all that. And the analysis which does that is called Kaplan-Meier. And if you don't have any cataract, it's a flat line across the top. So any decrement down from there means there's a cataract. So the percentages refer to how much of the lens was in the field. So if there was less than a quarter of the lens in the field, you only had a 20% chance of getting a cataract by our record. However, if you had more than 3/4 of the lens in the field, what happened was within 2 to 3 years, you don't get it right away, you get it within 2 to 3 years, a cataract appeared. Now this is not life-threatening, but I found it quite interesting, and I thought it would be interesting to another cohort, in particular NASA, because they have occupational exposures to particles in space, and I thought they would be concerned about that, so I wrote a few grants to them. And the whole science of studying the body in space is a new emerging field that I hope to excite you about, because there's much to be done. In National Geographic, again, you can guess I like that magazine, but the body is studied in space, and they review some of the things. For example, microgravity alone causes bone remineralization and other problems, but the radiation is always the showstopper for my interest. And when you're under an extravehicular activity, as the crew members often do for repairs or just maintenance of their vehicle, they're totally exposed in space to the radiation flux. So one of the first evidences that there was an issue of radiation effects in the eye were predicted by the man to whom I came to work with here at the lab, Cornelius Tobias. He reasoned, without having any evidence, that particles in space, which he was aware of, he predicted in 52 that travelers would see light flash phenomenon due to the traversal of those particles through the retina. And people thought he was crazy. But in the 70s, the American astronauts, while trying to sleep, were having trouble because of the light show they were having. And it was during solar activity when it was high. So actually, as a result of that, here at Berkeley with accelerators, several scientists here pursued that to prove that it was in fact related. And I'm going to show you some of that. These are several of the people, I think there were a total of 4. It included Dr. Tom Buttinger, Dr. Tobias, and the lab director at the time, Dr. McMillan. Now, you couldn't do this kind of science today. There are rules against this. But to their credit, I want you to know that they did this the first week I was here at the lab. And I thought, wow, they really believe in their calculations. They calculated exactly how many particles could be given to the eye, no more and no less. And they had to do it, a picture was just put together for your benefit, but they actually dark adapted them because the science of it wouldn't be valid unless you were dark adapted. So they wore dark glasses, and they did this study where they didn't know if the beam was on or not unless they clicked a little clicker to show that they saw a light. And the correlation really showed very nice work, and it's very impressive work, actually. The kinds of things they saw, depending on the angle of the traversal through the retina, look like these spots. Sometimes they saw hot dogs, sometimes they saw clouds. And so the whole concept of being able to see the radiation, and I've talked to astronauts myself, they say, it really makes you wonder, Ellie, because if it's going through my retina and making a light flash, what's it doing to the rest of my brain? And that's indeed the issue that NASA still has to square away. Now, what is a radiation cataract? Because obviously if the retina is having effects in such an exposure, you'd have to worry about cataract. So by definition, a cataract is an opacification, which means that rather than being crystal clear, you have a malfolding of the proteins there that it makes it not clear. And we had a large history with X-rays and gamma rays from the early days in the 60s that was summarized at Columbia University by Miriam and Folk. And they found a very interesting result that unlike most stochastic events, the appearance of a cataract was more deterministic. And in their language, that meant it had a threshold. That if you could sustain a dose of 2 gray and not get a cataract, up to 2 gray, beyond that, you would get a cataract. But now from our new work, we find that the dose limit actually does go down to lower than this limit. But let me just show you the old literature. This is complicated, I'm sorry. These points all mean that the doses were administered in a fractionated regime. So only draw your attention to this line here. There's the 2, and now it's in Rankin, so it's an old reference. This would be like 2 gray equivalent because photons don't have much of an effectiveness. If you notice, the spots are filled in above 2 gray, and below they're open, and that meant the open ones did not get a cataract. So, but you notice the data are widely scattered. The estimates of dose were not well controlled. It brings to mind the fact that many of you may not realize that all the different organs in your body have a different radiation sensitivity. And here in a linear array with an axis of centigrade up at the top shows you there are some organs like the lens which are highly radiosensitive, which is what I argued in my grant to NASA. I said, this is a very good dosimeter. There are other organs like bone which are more resistant. And in this presentation, the brain, I might add, was considered resistant because they were thinking there were not dividing cells, but now we know that there are indeed stem cells in the brain that divide, so this may have to be revised. So tissue effects all have a molecular basis, and that seems novel to some, but that's really the fact. And in general, radiation sensitivity of a given tissue, and therefore the whole organ, depends on the radiation-sensitive key cells in that system, the stem cells by and large. They're also important in radiation sensitivity are several parameters of the dose administration, and you have to keep that in mind. Now back to the lens. I'm taking the lens that you saw in the picture of the whole eye, and I've cut it in a section, and the light side is up here, and what you see is 2 cell types, a little square cuboidal type cell called an epithelial cell, and around the bend here at the bow, they elongate into lens fiber cells. So the lens as an organ only has 2 cell types, an epithelial cell and a fiber cell. Now, the light side, then you have to realize it's a whole disk of single cells. And the process of differentiation entails, in the final stages, a loss of the nuclei. You see that little dot in the middle of the square? It's lost on either side, and that's again a trick that the lens does to keep the lens crystalline. It completely abolishes its nucleus, its Golgi, its ER, the endoplasmic reticulum. All those internal organs are let go. They just disappear. They're chewed up by enzymes. And so much like the process we know is apoptosis, they are removed. And so that was a very interesting thing, because in the textbooks from medical school I found in the library, it said that the mechanism of radiation-induced cataract is damage to the lens epithelial cell. But at the point that radiation cataract is produced, it actually appears at only one location. It's called the posterior subcapsular location, the PSC cataract. And that's diagnostic for radiation exposure. You can get, if you have a diabetic cataract or some other kind of cataract, many different kinds of cataract. So the very unique anatomy here contributes to that story. Now back to my story. So I decided after all this work with the patients, there are certain questions you can't ask. So what we need is to be able to grow these cells in petri dishes in vitro. And I went to the medical school and I tried to find out how to do that, and they said nobody can do it. They try it from animals and only can get them to grow for a week or two, then they die away. Then I found a colleague, Jiming Louis and John Polanski, who said, you go to the slaughterhouse 3 times a year and you harvest, you buy the eyes that they're discarding from the meat production, and you harvest the cells inside the cornea, called the corneal endothelial cells. And my student group just did this last week, so you can ask them about the details. And using trypsin, we actually scrape away those cells and put them into petri dishes. And this is the arduous part. We grow them up for several weeks, and they have to be fed, taken care of. And then we lyse the bovine endothelial cell after we've added Textran, which is a trick to make them elaborate a very beautiful matrix. It's called extracellular matrix. And it's similar to the capsule that encases the lens. And this is a very nice theme in my own division of life sciences. My director, Mina Bissell, long has held that growing cells on naked petri dishes isn't the way, because the cells need the signaling from their environment. And it's really true, because I tried matrix from other parts of the body and it doesn't work. You have to make it from this position, because those cells make cytokines and growth factors that diffuse to the lens and allow the lens to grow. And there are also such factors from the retinal side that stimulate the production of the lens. When we do this in the lab, we get growth curves on the matrix. And the curve, the plot here on the left, the figure on the left, is not a tissue section. Those are how the cells look after many weeks of growth, 3 or 4 weeks of growth. What you see are the blue dappy nuclei are the DNA, and they've started to elongate before they start to disappear. The red filaments of actin are stained with phalloidin, and they fluoresce red, so you can see them, and that they're getting parallel, just as you saw in the whole organ. In contrast, if you take a cell that has been immortalized with a virus that will grow on plastic petri dish, the morphology is quite different. It's not normal. The DNA is all circular, and if you're studying actin in the transformed cell, you're in trouble. Now, we've done a lot of different kinds of experiments with these cells in this model. It's very unique because it's hard in an animal to harvest the cells at the bow that are doing this transformation and differentiation. So I'm just trying to, in this very short presentation, give you an overview of where we've gone. And when you irradiate, there is a sensor system. You have certain genes that are very responsive to DNA damage. And they trigger a signal transduction pathway and a cascade to effectors, including such things as P21. And these effectors are what affect differentiation, cell cycle, cell viability. Now, I've already told you that the different tracks do different things. Well, if you look at carbon and alpha and protons of different energy and different LET, you do see different effects. And of course, depending on the anatomy of the DNA at the time of the exposure, you'll see different things too. But if you take DNA and unwind it so it's a flat presentation, like my colleagues Holly and Chatterjee here at the lab do in their theoretical modeling, They do Monte Carlo modeling of tracks through DNA in this presentation, and the solid circles represent sugar damage to the DNA, and the squares are base damage. Oh, sorry. And what you see is if you have the low LET particles, there's sparse damage, but what those big fat tracks do is quite a bit of damage at the high LET. Now, one of the things that has to happen when a cell decides to differentiate is it has to stop in its cell cycle. And that happens between the phases of G1 and S phase. And there are a family of 3 different families of cyclin-dependent kinase inhibitors that achieve that. And P21, which I have some interest in, is one of those. P21 has a long history of study in cell cycle. It also has a lot of people studying it because of the stress response that it is upregulated after radiation damage. It gets activated. But the interest in differentiation is sparse because there are very few people who can actually see differentiation in culture. Now the Germans have accelerators, and they have actually traversed nuclei of cells. The red, here the DNA instead of being blue is red. And what are the yellow spots? Well, that's an antibody to P21. And what this illustrates very graphically is that this was taken minutes after an exposure to lead ions at 3 by 6 centimeters squared, 10⁻². And you can actually see where the damage happened, and the P21 protein is recruited to that damaged portion of the DNA. And they actually did fiduciary work to confirm that it correlated with the exact location of each traversal. So, but with regard to terminal differentiation in P21, there's much less known. When I went to the literature, when we first started with the lens, I found description of P21 and terminal differentiation of crypt, keratinocytes, skeletal muscle, hematopoietic stem cells, fat cells, monocytes, but nobody reported P21 in the lens. What I did find was that people were reporting other members of the family, the CDKI family of the P21 CYP-KIP, called P57 and P27. And this is primarily in work that was done in studying embryonic development of the lens. The only place they saw P21 is if they made a transgene, which had genes with E2F1 and E2F2 modified. So my lab has been engaged in looking at the P21, 27, and 57 story at both the RNA and the protein level for some time, and without radiation, because we're very interested in the normal process of differentiation of this epithelial cell. And it's important because you realize most cancer arises from defects in epithelial cells. It was obvious from our protein work that 27 and 57 showed major changes as you went from epithelial to fiber cell, which explained why people saw that. The P21 increase was much less obvious. But when we used the P21 red antibody, I'm sorry to keep changing the color, but this one is red. And if you look at the left panels, which are not irradiated, the epithelial cohort here and the differentiating cohort here are different. You see blue nuclei primarily in the epithelial cohort with 2 little pink nuclei. And if you overlay red on blue, you get pink. And that meant that those cells are starting to differentiate. Whereas in the more confluent culture, you see low levels and very heterogeneous expression of P21. However, in the stress response, after 6 hours after 4 gray of X-ray, you can see the epithelial cohorts all show intense pink color, and the fiber cells also have an upregulation. That was at 4 gray, so we've now taken it down to lower doses that actually might be encountered in space, and we still see it's more subtle, but there's still an upregulation. And that also is true in the differentiating cells. So then the question is, are we only finding it in the model, and is it not true in the human eye? And I have a colleague at Harvard Med School, Dr. Lee Goldstein, and he kindly had donor eyes offered to him that he offered to me if he did the sectioning of them actually. And I sent him my antibody. I don't know if you can see it. There's a trouble in vivo because the lens has autofluorescence, which happens to be red. So if you look here, do you see a red sparkly color? That's the P21 positive. And this is at the bow, right here at the corner of the lens. The anterior central region up here does not show that sparkly. So the P21 expression begins as the differentiation occurs at the bow of the lens. And he confirmed it. We're just writing this up. Now P21 is a very promiscuous protein. Maybe why it's so important. Its amino end binds certain proteins, and its carboxyl end binds other proteins, and they're very important proteins. So Dr. Polly Chang in my lab has been doing quantitative real-time RT-PCR, and this shows some of her, just an example of some of her work after X-ray, where we're looking in the epithelial and the fiber cells at a time course of, after a single dose of 4-gray, you see this kind of an expression pattern in the summation of a couple of experiments. There are waves of expression of P21, which we think is miscuing the differentiating cells. Well, I don't have time to show you everything, but in this compilation, the pink and the yellow are protons at X-ray, and we don't see much difference in the P21 changes, although we do see this many-fold increase. I might draw your attention to this lone blue triangle. Because we no longer had iron ions here at Berkeley when we were doing this, we had to travel to Brookhaven National Lab where we did an iron ion experiment. And as you might guess, iron ion might be very important to P21 response because we see this huge effect, 10-fold increase in comparison to the proton and X-ray result. So in space, most of the radiation you have is protons, and there are only a few iron ions, but the iron ions may be very effective, and so we're trying to understand this. Most recently, Polly's gone on to do functional gene analysis, looking at more than just this family, and she has shared with me this gene array study, where you have a filter that's blotted with a whole large number of genes. And there are controls looking at housekeeping to make sure everything is in order. And by and large, after we did our studies, we were able to confirm that we saw an upregulation of P21 and a downregulation of another whole set of genes. So we're just piecing this story together with a hypothesis that I'm going to share with you shortly. So just to summarize the tissue culture work, our results suggest that P21 expression in our in vitro differentiating lens cell model parallels the pattern of normal differentiation seen in adult lens tissue. All of us right now are making lens cells. It's one of the few organs where you actually have that through adult life. Now, with radiation, there's a triggering of upregulation of P21. Now, if the cell thinks, if I have P21 going up, I should be differentiating, we think the radiation is prematurely having the cell think he should start differentiating. And so we also see a similar pattern after X-rays, and our preliminary iron data are very suspicious but unconfirmed yet. But it could be a much bigger effect. So the magnitude of these transcriptional induction changes suggest a dependence on the dose and stage of the differentiation. And our model, this little cartoon, shows you that the cells, as they migrate toward the bow, are beautifully lined up in parallel. And then in the last stages of terminal differentiation, they elongate and enucleate. The nucleus disappears, and that maintains a crystalline lens. And during the course of that, there were molecular hallmarks which we can monitor in the laboratory now in vitro, which this big, slow triangle shows you of all these genes, a whole complement of proteins and genes that are changing, whereas there are reciprocal decreases in other things. Now, in contrast, after we irradiate, things are a little bit different. The cells, instead of being beautiful in parallel, they start showing bad alignment and faulty enucleation, and it's also goes along with a very different profile of the gene expression. We see an abrupt increase and abnormal levels of these proteins. And we see also abnormal cyclin-dependent kinases. So we think that's the mechanism for radiation-induced cataractogenesis, and we're still working on that to prove it. Now, what does this have to do with astronauts? Well, NASA funded this work for several years before they thought, well, maybe we should check and see what the astronauts actually have. So my program manager, Dr. Frank Cucinato at JSC in Houston, was the author of this paper that came out in the year 2001, where they actually looked at the astronauts' career exposures to radiation. And this is a plot that's not often seen. Let me orient you. Every number here refers to an astronaut. So it's nearly 700 individuals. And there are a couple of dots. The triangle at the bottom is what their badge read, which is uncorrected for effectiveness. And the circles above, for each of them, they show a different effectiveness depending on where they were in the spacecraft. So what this is actually where they take astronaut number 1 has the lowest career dose, and astronaut 660, he has the highest. So this is a distribution showing the increase. And what it illustrates in units of millisievert is that when John Glenn first went around Mercury a couple times or once, he didn't get much dose. It was less than a millisievert. But now with Apollo and on to Skylab, and now when Mir was up, and Space Station and International Space Station, the doses are 100-fold more. And so NASA has an issue to settle here. They need to understand that in further study and exploration, the doses, will go up. Now, what has been done, each of them are well badged, and each astronaut, and you understand what their lens doses are. And in particular, the cosmonauts and astronauts on Mir sustained pretty significant millisievert doses, but certainly well below the thresholds that everyone was thinking existed. But you have to realize that we're not really sure what the incidence is due to, and we actually do see cataracts. These are cataract plots like I showed you for the patients who were treated for uveal melanoma who got a cataract. It's a similar type presentation where the flat line at the top would mean no cataract. What they did was they took the astronauts and divided them into two cohorts, whether they had an average low dose or a high dose. And I say high with quotation marks because it's only 45 millisievert, which is pretty low dose. And yet, whether they scored all cataracts or just the non-trace cataracts, it's clear that even the low-dose astronauts show incidence of cataract much before their age would have expected. And the high-dose ones, 2, in fact 4, have gone to surgery. Now, predictions for Mars. It's sort of daunting. The Mir data are here, and the predictions for Mars, without additional information on shielding and countermeasures, are quite extraordinarily high. We're hoping this isn't going to be a showstopper for the exploration of Mars, and that's one of the things that we're working very hard to study. Now, before closing, I want you to be aware that I've only restricted my remarks to the discussion of radiation-induced cataract. Radiation does a lot of different kinds of things to the human body. The one kind of late effect that is have major concern is cancer induction. The astronauts all want to know, am I going to have a higher incidence of cataract as a result of my work in space? But there are also concerns about neurodegeneration. And I want to just close with a few new studies that we're launching into in that regard. When I met Dr. Goldstein at Harvard and he learned about my model after helping me with my P21 study, he asked me if I could use my model to help him with his work. So I said, what kind of work are you doing? And he said, I'm studying Alzheimer's disease. And this is a cover of a magazine where the brown plaques associated with the beta amyloid dementia in Alzheimer's disease is illustrated in a cross-section of a human brain. In this normal brain, you don't see the plaques, and again, on the Alzheimer's side, we call it, for Alzheimer's disease, we call it AD. The AD brain shows the brown plaques, and you also see significant loss of brain material, which is really of concern. Now, the beta amyloid is usually studied into the membrane in a precursor protein called APP. Just a little chemistry here. Biochemistry, and there are proteases like BASE that cleave this molecule and release, and there are cleaving molecules on this side, the gamma secretase, that actually release the A beta in a mechanism we don't know. This is a cartoon that shows our ignorance, but this is what we understand. And these A beta tangles actually outside of the neurons are associated with insoluble proteins, and that's what makes the brown plaque. Now, Dr. Goldstein explained to me the reason he wanted to work with me is he was just about to publish a paper, which he did in The Lancet in April of this year, in which he found from his clinical experience that all Alzheimer's patients also get a cataract. And the cataract, the actual opacification was due to beta amyloid. And so he published this very remarkable finding in this citation. And previous to his work, the AD patient is considered only to have beta amyloid in the brain. It was never found in any other organ of the body. So this was quite striking. And what he asked me to be involved in was a study in using our normal lens differentiating model to investigate this further. So, but let me just tell you a little bit about his work. He had 9 donors with AD and 9 normal age-matched controls and sex-matched controls as well. And he did this whole array of different studies to characterize And these are some of the lenses, and I just want to, I know you're probably not familiar with looking at cataracts, but I just thought I'd illustrate this for you. Most ophthalmologists use a technique called slit lamp, which gives you this yellow stripe. And the reason they do is where there is a cataract, you get light scatter off the opacification, and then it makes it very obvious. But I find most people like to just look at the lens and see where the opacification is natively first, then they can understand about the scatter. None of his control non-AD patients had a cataract. He also had among the elderly donors, and these are people who very graciously after they died donated their eyes. We can learn a lot from people who do that. These people, one of them had a Parkinson's disease, which is also associated with dementia, and yet it didn't have a cataract. So we believe that the cataract from the AD patient is very particular. In fact, it's called a supranuclear cataract. So unlike the posterior subcapsular, the supranuclear cataract is very perhaps diagnostic. And this would be a big, huge advantage because you may not realize that most clinicians, if you bring a loved one to them and ask them, does my loved one have Alzheimer's disease, he really can't tell until he does the pathology on the brain, which of course doesn't help you much, nor the patient. But if we have an indicator like this, it would be very important. And it turned out that they decided to make a transgenic mouse because mice don't normally get AD, only people. But if you put in a transgene for A beta, they do get Alzheimer's disease, and they also get this cataract. And what they've learned from the animal work is that the cataract appears way before the dementia. And so if we can diagnostically use this tool, we may be able to intervene. Here, Lee actually did mass spec and proved that it was the beta. He also showed it was in the retina. So his interpretation in the bottom line of his work that was published is that the beta amyloid is present in the cytosol of the lens fiber cells where this peptide may promote regionally specific lens aggregation, translate that into cataract, and is specifically a supranuclear cataract. Now, I told you about the mouse model. Well, people thought, well, if it's the plaque that's the problem, we'll just make a vaccine and get rid of the plaque. We'll fix that problem. But when they applied it to the animal, the red plaques here, the animals given the vaccine did indeed have fewer plaques, and everything seemed okay. But when they took this to human trial, they had to curtail it after the first patient died. So what it speaks to is the possibility that A beta has a normal function in the lens that we need to really understand before you start using vaccines to eliminate it. So now to our model. These are immunofluorescence pictures. I probably should have a black background, I'm sorry. They're 3-day, 7-day, and 10-day cultures, and the red color is the beta amyloid. And what you see in the early cultures, and the cells are epithelial, is it's nuclear. At 7 and 10 days, the red antibody goes outside the nucleus into the cytoplasm. And he thinks that's very significant that there's a processing in the normal, this is not irradiated, these are normal lens cells. And the kind of processing I'm talking about is the APP molecule, if it's cleaved at different places, create different carboxyl terminal fragments, either a beta or an alpha. And so he wanted to do some Western analysis. So we harvested proteins from cultures at different ages, and we definitely see the mature and immature form of APP in our model very cleanly. And in the epithelial cell, we see in this overexposed version, you see that the beta CTF, this is the beta CTF fragment, which is not seen in any other model. By the way, they tried the immortalized human lens cell, and they didn't see it. So it's only the non-immortalized human cells that show it. So I'm at the end of my story. It's time for me to quit, but I just thought I'd give you a summation of what I've told you. The science of particles and radiation and understanding how different kinds of radiation interact with tissues actually works very well with the invention of Ernest Lawrence and the intervention of his brother John in the use of the cyclotron for treating cancer patients. And that work, there's a long list of people I have neglected to mention. Dr. Joseph Castro, Robert Wilson, who had the idea, and many others, Ted Phillips. And they treated cancer patients for many years, more than 15 years here at the lab, with some measures of success. One of the spin-offs was a study of radiation cataract, which I was involved in, which led me to try to grow human lens cells by tricks we now have available to us using extracellular matrix. And this allows us now to really have a molecular basis to explore what's going on in the laboratory. And so the coincidental appearance of cataracts in the in the astronauts is very significant. I should mention that at Berkeley, when they had ions here, many studies were done with animals, which I've neglected to speak to you about. I've restricted my remarks to human studies. The reason is a mouse only lives a couple years, and the human cataract appears in 2 years. So each species has a quite different presentation of the cataract. And there are questions about species specificity that our human cell doesn't have as a problem. So in summary, our HZE radiations have unique physical deposition patterns and some novel characteristics of the biological response depending on the radiation type and the radiation quality. There's definitely a need for further basic biological investigations to clarify the significance of these unique lesions at all levels, molecular, cellular, and tissue. And there are many powerful new techniques available to us, looking at genomic and proteomic resources to study these effects. And also, significantly, we need further modeling because we can never do all the studies we need to do. I also would like to show you in conclusion that NASA has moved cataract up. They have an engineering point of view, NASA. You know, if you want to go to Mars, you make a list of what you have to do and put priorities on them. Well, cataract used to be down here number 4 priority as a radiation risk hazard. But as a result of the work of Frank Cucinato and his studies of the astronauts, it's been bumped up just below cancer for further study. And there are resources from NASA to support that work. And finally, I'd just like to acknowledge my laboratory and some of the people who helped me prepare this presentation, including Chris Rosen and Becca and Karen Odo. And the scientists in my group, Kathy Bjornstad and Morgan McNamara and Chris and Polly Chang, who have done many of the studies, the results of which I've shown you. Our collaboration with the Harvard group and Brigham and Women's and Ron Mancini. Thank you very much.
Just wondering if you have any modeling or direct experimental results that would identify any significant biological effects to the eye as a result of near space or higher altitude exposure, either during ambient conditions or solar proton events.
Yes, you ask an important question. Actually, astronauts are not the only ones who get these particles. When we take an aircraft and fly, I often see those 100,000-mile flyers getting on. I'm thinking, are you sure you want to do that? Actually, but the dose exposure level is microsieverts, so it's significantly lower. And if you're flying on the equator, where you don't have the defect in the Earth's magnetic field, then it's not an issue. But it has become quite politically handled in our country. Our airline industry lobby is quite strong. The Europeans have actually badged people and consider all airline crew radiation workers in Europe. And they're concerned about the pilots, especially that fly Norwegian routes or polar routes to Japan. And I have another whole lecture I could give you on that story. It's very interesting. And they usually work 600 block hours, each pilot. And now the pilots are concerned too, so they're actually having biodosimetry done to look for sensitivities, because one thing I failed to mention to you is if you have radiation sensitivity, it's been shown that you also have increased incidence of radiation-induced cataract. So I don't think I've answered your question exactly, but it's well under study now by the FAA.
Another question? Okay.
Yes, I was just wondering if you could speak to how difficult it would be to shield a spacecraft or even a spacesuit against different types of radiation.
Yes, you ask another good question. The only problem is particles undergo a process called fragmentation. So if a particle comes in and hits like an aluminum shielding, it actually fragments into an array of particles of lower atomic number. So you actually have a higher fluence on the inside than you would have on the outside. So there's been recent study by NASA of the materials of the spacecraft, because hydrogenous materials like shielding, polyethylene shielding, can reduce just by the different Z of the impact of the ions coming in from space. So shielding has limitations from that point of view. However, I talked to an astronaut that his vision is that, of course, what you want to do is minimize the exposure to Mars. So he's a big proponent of alternative propulsion. And he would like to put the propulsion material, which is hydrogenous, in big tanks around the spacecraft. Now, that would really ruin your view, but it certainly would shield you. And so they have lots of things under study just to examine different alternative shielding. But the biologists have an alternative approach. They are looking at nutritional basis, because if you're a high meat eater, you make oxidative products in a greater way. So most of the astronauts on these long flights will be vegetarian. And they have other countermeasures, nutritional countermeasures of antioxidants, including vitamins, which are already established in the clinical world to protect against some of these effects.
On these hypothetical models for the trip to Mars, what about solar storms?
Yes. They have, in certain of the designs, initially decided they wanted to make a storm shelter where the astronauts could recede to if they were detecting a solar event. But as I've already discussed about the fragmentation and shielding issues, it's not clear that how much protection that would afford. And so for some of the missions, until they reach a certain distance away from Earth, they might bring them back, but there are limitations on orbits to do that. So, and we have very poor prediction for the solar storms. It's a 50-50 deal. The best in the field have told me 50-50. So they really have a lot of theoretical work. There are solar cycles, I didn't show you that have minimum and maximum periods. And so what they are planning is to strategically arrange the orbit to have minimal dose from solar expectation, even with its limitation. And the character of each solar event is quite different, whether it's more mixed proton or heavy ion.
Okay, thank you.
Well, thanks again, Ellie, for an excellent presentation.